Hi, my name is Divya Dar. I'm the business lead for MTAM, which stands for microtube array technology. We've created a membrane that is 1/50th the thickness of our closest competitor. With this micromembrane, we're able to detect the best drugs for cancer detection and allowing us to fight cancer. So how does this work? The problem in drug development is that it's full of very long life cycle. It's very arduous, very expensive. It takes about 100,000 compounds for leads to then be converted into one FDA drug-approved drug. And because of that, the response rate amongst patients less than 40%. MTAM can help at every single part of this product life cycle, from screening to trial to personalized medicine, enabling us to do drug selection right at the patient's bedside. So how does this work? MTAM is refined a technology that is more than 30 years old, hollow fiber assays. This is an animal model testing technology. Because our micromembrane is 1/50th the size of our competitors, we create results that are much more reliable, 1/4 the speed, and 1/100th the cost. This allows us to target personalized medicine as the first market, enabling us to target a market potential of more than $2.5 billion in the US alone. We have a remarkable team with our technical lead, Dr. Chen, who is an associate professor at Taiwan. He has more than 30 years experience in material science, more than 20 patents and 80 publications. I'm the business side. I'm an MD and an MBA, and I've led a company through to exit with my last company in health tech. We're using MTAM to fight cancer. In the last 2 months, we've added the business side. We have more than 8 patents. We're targeting 2 specific business sides, which is personalized medicine and drug trials. We've got over 5 customers with early revenue of about $300,000. We have about 5, we're doing clinical trials around 5 sites with 5 sites with 200 patients. And we're using all of this to show that our technology can predict drugs. We're seeing positive results of more than 90% predictability. To give you an example, our latest clinical trial has shown that we were able to detect PD-1 immunotherapy drug to a high level of predictability in lung cancer patients. We predicted the drug, the doctor ended up using it, and this led to marked tumor reduction for the patient in just 2 months. This is just the beginning. We're here asking for $3 million to help us set up a US infrastructure and facility, start clinical trials. We believe our patients deserve empirical evidence before drugs are given to them. That's what we're here to do. We hope you'll join us. Thank you.
Great job. Any questions for MTAM?
I'm sorry, I'm just a little confused. I thought at first that you had these tubes that that you would use for drug screening early on, and then you got into this whole combination therapy selection, and I'm not really sure what you do in that. Can you clarify a little bit?
Absolutely.
Or do you have like 2 or 3 separate businesses?
Yeah, because what we can do with our micromembrane technology, it's basically a platform type of technology. You can use it both for personalized medicine, as in treatment selection, but also in terms of drug selection in the earlier part of clinical trials. The way that the technology works is that there are microtubes in which you can insert human cells, which are tumor cells. You can use that and put it into animals such as mice, give the mice the drug that you would imagine giving a human, and that allows us to show which human cells ended up dying because of this drug. And then you can use that, as you can imagine, both on the clinical side with drug dosage, but also on the pharma side with enabling them to figure out which compounds are the best to push forward.
So if you drive this to further down the line, would you imagine that as part of, say, an immune therapy selection, that for each patient, you would conduct an animal trial, like during the first 10 days?
Yes,
of
being
the
a
treatment?
physician, I think that should be what's happening right now. Right now, majority of drugs are being selected almost with like no evidence. It's just more based on guesswork, and it's, I think, very irresponsible. I think ideally what you say is like, we believe these are 2 or 3 best drugs available for this patient. Let's do a 10-day, it only takes us 10 days to figure out which is the right drug, which is perfect timing for a cancer patient, and then you can give them that instead of, instead of all the complications that can be led from these kind of drugs.
Okay, and then roughly what would that cost for one patient?
We think it will be around $3,000, which is about 1/100th the price to 1/20th the price of the competitors.
That includes the rabbit?
Exactly. But we're just using normal mice.
Cool.